Hi, in this video I'm going to explain how scientists find locations of the genes, and here's a problem. You are trying to locate an enzyme producing gene in Drosophila, which you know is located on the third chromosome. You have 5 strains with deletions for different regions on the third chromosome, and this sign indicates a deleted region. So here's a table. You cross each strain with wild-type flies and measure the amount of enzyme in F1 progeny. The results appear below in what region is the gene located. And even if you don't know how to solve this problem, you still may try to solve this problem using common sense and logic. So I recommend you to stop video here, try to solve this problem, and when you would be ready you can run video again and you can compare your answer with my answer and explanation. So let's first analyze this table, and as you see, strain A shows 100% of the production of enzyme, and that means that here this deletion doesn't affect production of the gene of the enzyme, and that means that this deletion not in the gene region. So we can cross out this deletion, and also we can shadow not only this deletion, but we can continue on the other strains as well. And for example, strain E show even better results than A, and this is also normal because you can never get the same results. There is small fluctuations, and it also means normal results, and that means that this region here in strain E doesn't affect a gene, and we can shadow not only this area but also area of other strains, so this area doesn't contain a gene. And strain D also shows good results, and those it's a little bit smaller than E, but it's still normal fluctuations in production of enzyme, and we can also say that this deletion doesn't affect a gene, so we can cross out and shadow all this region over all the strains. So what we left with strain B and C, and as you see there is a great reduction in production of enzyme, and that means because flies are diploid and Drosophila has 4 pairs of chromosome, and for example if we take chromosome, for example number 1, one would be from the mother side, another from the father side, and for example this one can have defective gene with deletion within a gene, and it wouldn't produce enzyme, or enzyme with such a deletion wouldn't function properly, so this would produce proteins that wouldn't function, and this other gene here would be expressed and would produce normal proteins that would function, but as you see the number of them would be reduced, so we would see reduction in the amount of enzyme produced. So as you see, deletion in strain B means reduction, and in strain C also reduction in production of enzyme. So this deletion affects enzyme production, and this also. And if you think that gene located here where two deletions overlap, this is not so. We can put the borders here that we know for sure that these deletions don't affect gene and enzyme production, and deletions here also don't affect enzyme productions, but this deletion and this deletion does affect. For example, the gene can be located somewhere here. It can start here and end here, or vice versa. It can be here and it can go in this way. It even can be shorter. For example, it can be only this large or this small, and we only know for sure that it should be 100% where we have overlap, but we cannot tell for sure how big this gene. So this is going to be our answer. We only can tell for sure that the gene should be in this region, and we also can say for sure with 100% that here where we have this overlap, the gene would be present in both strains, but we are not so sure about these regions and this one. This would be under the question, and nowadays with the use of computers and bioinformatics, such information would be enough to search for the homologous genes and sequence in other strains, so we can correlate information about known genes with unknown sequence, and this is how genes are located. And I also want to add that this is not the only one way how scientists can locate genes on the chromosomes. There exist many other different methods, and this is just one of them. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.